years, our families had a share of health problems.
So prescriptions are a part of our life.
Before we went to the big box store.
We both thought it would help us save. But with the long lines and impersonal service, filling prescriptions became a chore. That's when a friend recommended D&H.
Now Tristan knows our prescriptions. Brenda always helps us find the right vitamins. And after Dad's fall, Monica's been a real expert with all our home medical needs. All without the lines.
Trust and service.
That's our D&H.
Welcome to Radio Friends on Thursday, December the 17th. Got a really interesting guy here today. Been on Radio Friends several times, and actually we just pick a topic and he goes. Jack Schultz, Director of Life Science Center at University. Good to have you here, Jack.
Good morning, Paul. It's great to be here.
When you came in today, you said you want to talk about CRISPR. And I said, what CRISPR? You mean like a CRISPR in the refrigerator?
No. Well, it's an acronym for a much longer science term, you know. It's the kind of thing that we'll never remember, so just think of CRISPR.
Okay, CRISPR.
Okay. All right. Pardon me. It refers to a way of editing genes, which means you can find a gene in some buddies or some plants or some animals' DNA and very precisely, quickly, and efficiently snip it out and replace it with a different gene. Now, we do that already with various kinds of techniques. In fact, one of the important techniques was discovered in a hot spring in Yellowstone National Park. You ever been to Yellowstone National Park?
No, I haven't.
Oh, you may have seen these beautiful blue and green pools. They're hot. They're boiling water.
I've seen pictures.
Okay. The colors come from bacteria growing around the inside of the pool. And what's unique about those bacteria is that they can live at such a high temperature. And we call them extremophiles. They like extreme places. If you think about it for a minute, though, in order to live there at that temperature, everything their cells do has to work at boiling temperatures. I assure you that none of our cells, yours or mine, will work at boiling temperatures. The proteins fall apart. It just doesn't work. So people have discovered lots of things that those bacterial cells do that we can use if you heat them up. And so you can do lots of interesting experiments where you use bacterial tricks for slicing DNA, making proteins, all those kinds of things that cells need to do at high temperatures. And the benefit of that is that you can turn it on by turning the temperature up and turn it off by turning the temperature down. Yeah, so it's very controllable. Well, this new technique is sort of like that. It happens at room temperature. And there are a lot of details involved, but what's really cool about it is that it has a very precise way of finding even a small section of a gene and carries along with it like a pair of scissors to snip that little piece of the gene out. So you can do a couple of kinds of things. One is you can replace genes or you can remove them. The reason you would want to remove a gene would be, for example, to see whether that gene really does what you think it does. So if we're studying plants, I see we have some lovely poinsettias here. If we have a guess about what makes the poinsettia red, we think that's this gene over here is probably involved. We can snip that gene out, and if they're not red, we have pretty much shown that gene was responsible for the red. Okay, this new tech, that's kind of sloppy and it's slow. It kind of takes a long time to do that. And if you're working with animals like, the lab rat or mice, that sort of thing, they take a long time to grow up, reproduce. You have to mate them and get, even if you've got a gene snipped out, you don't see the result right away. You've got to get it into the babies and then see the next generation, all that. It takes a year or two to create a mouse that is missing a gene you want to study. What's a good example? Well, we have investigators in the Bond Life Sciences Center who remove genes that they think are important for proper nervous system development to see whether the mouse actually has some sort of neural defect when that gene is gone. And that's very useful because now you know where to go to look for cures.
And you can, if you know that's a problem, you can replace that defective gene
That's right.
with a gene that does not
works.
have the neuron.
You're right. And the most convenient or the most likely way to do that now is by putting that gene into a virus and infecting the person with the virus, because viruses insert their genes into the genomes, into the DNA of their hosts. So you can use a virus to deliver a new gene. The problem with that is that may not be the only thing a virus does. And attempts to make this work have sometimes run into the problem that the virus itself has negative impacts on the person or the host, even if it fixes that one gene. So it's been in development for years and never succeeded that much.
What you were telling me when you came in is that scientists now can clip a gene or insert a gene and change the color of your eye.
Mm-hmm.
It can change characteristics that you have?
Well, not yours, but
Right.
your offspring's. Your offspring. You have to do this on unfertilized eggs. Okay, very, very early in development.
Can you clip a gene that would cause cancer?
Absolutely. And this new technique, the one called CRISPR, is getting a lot of attention. It's been all over the news in the last couple of weeks that scientists are now they think they will be able to correct some very important genetic defects that lead to important diseases. We have a really good example in the Life Sciences Center. Dr. Lorson works on a disease of children in which the nerve connections to muscles in the back they don't come together correctly. And that's lethal in children before the age of 3 or 4. Using the virus trick, he can now replace the defective gene with a functioning gene in mice, and the mice are fine.
Mm-hmm.
Okay?
So that could be used in people.
In a positive,
But then
yeah.
you have, in some aspects, you have moral issues with dealing with this.
Yeah, the thing, that virus procedure is risky enough and slow enough that it hasn't gotten a lot of traction. People are working on it, and some are pretty successful. But this new method allows anybody to edit genes very quickly and very easily.
Do you ever see this being used to totally eradicate some of the worst diseases that we fear?
Well, that's the question. And last month, scientists met in Boston to argue over how far they can allow them to go. And, it was a long argument about, well, here's the good, but there's the bad. We really don't want people editing babies to turn out the way they want them to look or act or something. But boy, the promise of curing important diseases is really there, and it's so convenient now that people are very concerned about it.
But as you said, there's also the negative side that you could go in and edit a human being and create the human being with the characteristics that you want, which could be good or bad too.
That's right. And you do this now in one generation. You don't have to breed mice for a year or something to get this.
It's interesting, Jack Schultz, and we could go on with you for a long time. The term is called?
CRISPR.
Okay. If they want more information, they can Google that, right?
You sure can, yeah.
All right. Jack Schultz, Director of Life Science Center. Always a pleasure to have you here. Please come back again.
It's a pleasure.
All right. Tomorrow, Heather Harlan. She's got a holiday story for us. Our program, directed by Travis McMillen of the Reynolds Journalism Institute. Audio is Pat Akers from KBIA. Our floor director is Kevin Casson. And our assistant producer and guest coordinator, Uncle James Mauser. If there's something you'd like to see or hear, I would love to hear from you. Drop me an email at pepperpmissouri.edu. If you're going to be out driving about, be careful. We'll see you tomorrow. Bye-bye.